Uh, Hello everybody, I'm Barsha Tripathi. I'm serving as Assistant Professor of Zoology in BJB Autonomous College, and for today I'll be taking the chapter Health and Disease. So for today we are just going to discuss what is health, then we'll talk about immune system and immunity, and then we'll talk about next communicable and non-communicable diseases. Then we'll talk about adolescence and vaccines. Okay. So for today, we'll start with health. So what is health? So good health is actually maintained by a balanced diet, a good routine habit, a sense of hygiene, personal exercise, physical exercise, and mental well-being. So good health can be disrupted by many agents, one of them being pathogens. Now what are pathogens? Pathogens are infectious agents. So let me write here. Pathogens. Pathogens are infectious agents that actually disrupt the normal physiology of the organisms, and they cause symptoms. So there are many classes of pathogens. Most important of them are, let me take another pen. Yeah. So these are prions, which are basically proteinaceous in nature. They lack nucleic acid, and the disease that they cause, that is infectious in nature. Next are viruses. Viruses are actually, they are at the cusp of living and non-living organisms. So when they are outside the body, they are non-living, they cannot replicate. To replicate themselves, they need a host. Okay. So the examples of viruses would be retroviruses, then we have picornavirus. So next comes bacteria. Bacteria are prokaryotic organism. So these bacteria, often gram-negative and gram-positive bacteria, both cause diseases. So the common pathogenic bacteria would be Mycobacterium, Salmonella, now next comes fungus. Okay, so fungi would be a would come in your eukaryotic pathogen. Then comes your parasites. So parasites, these could either be protozoan in nature, or these could be helminth. Okay, so these are the different classes of parasites that we have. So parasite is usually described in the terms of its ability to produce toxins, which are nothing but substances that would evoke immune response, their ability to enter tissues, colonize and consume host nutrients for their growth, and then their ability to induce immune suppression. So pathogens would always try to get away with the crime. Okay, they would like to not be detected by the immune system. Okay, so now having said all these terms, so there is one important term I should mention, which is the pathogenicity. Pathogenicity or virulence. Okay, so this is nothing but the disease causing property of pathogens. Now about diseases, that we will discuss in the next class. So we'll take up, so now that the pathogens, we are saying pathogens can have the ability to enter the body. Now what does our body do? So definitely it would actually come up with some sort of defense mechanism by which it can combat the incoming evading pathogens. So this defense is carried out by our immune system, and the response evoked against the pathogen is nothing but our immunity. Okay, so let's discuss immune system in details. Okay. Let's talk about immunity. Now, immunity refers to all the reactions exhibited by the body to inhibit, inactivate, or destroy the invading pathogens and their toxic products or other foreign substances that enter the body. Okay. So basically, immunity is nothing but our body's way of combating the the pathogens. Now, we can classify immunity into two classes. So immunity would be innate immunity, okay, and it could be acquired immunity. Now, what do we mean by innate and acquired immunity? Okay, let's talk about them one by one. Okay, let's first take innate immunity. Okay, yeah. So innate, the term innate means that when the child is born, the immunity, it is born with that immunity. Okay, so it is present from the birth of the child. That is why it is also referred to as inborn or natural immunity. Now, so this we write down the points. This is like, then comes the specificity. Now innate immunity has a low specificity, which basically means that it cannot distinguish one pathogen from another pathogen. But the reaction that innate immunity would go for is basically a very generalized. Yeah. So the next property of innate immunity, it is non-specific. So yeah. So basically, once the pathogen tries to invade the body, this is actually the early line of defense against the pathogen. Okay. So there are many, so when we talk about innate immunity, there are many components that are a part of it. So we'll discuss all of those components individually. Let me modify this to innate immunity. So, so the very first component are mechanical barriers. Mechanical or physical barriers. So these actually make the skin impermeable to microorganisms or pathogens, so that because of which the organisms, microorganisms or pathogens would not be able to enter the body. So these actually act as the first line of defense. Okay. So any incoming pathogen would have to first cross the mechanical or physical barrier to actually get inside the body. So what are the different physical barriers that are present? So the first one is actually skin. Okay. Now skin has epidermis, right? The outermost layer of the skin is epidermis. Now epidermis actually right here. Epidermis actually contains a lot of keratin. Okay. It has a layer of keratin. Keratin is nothing but a protein which is very tough and water insoluble. Okay, it's fibrous in nature. So what exactly happens is that this actually inhibits the penetration of any incoming pathogen into the body. Okay. Then the next point is that why is skin a mechanical barrier? So the first thing is the presence of keratin that makes the skin impermeable. The next is desquamation. So desquamation is nothing but skin epithelium. So the outermost layer of the skin is periodically shed. So what happens by this process is that any pathogen which would be clinging to the skin would actually be casted off. It will be shed off. Next is now skin actually lacks blood vessels. So what happens is that, and it cannot retain moisture. So all of these points actually make the skin an inhospitable environment for the pathogens to either enter or to survive there. Okay. So the thing is, let's just say a pathogen is able to cling to the surface, okay, but then it before penetration it would need to spend some time on the skin. So this actually, these mechanisms would ensure that the organisms is not able to, the microorganism is not able to survive on the surface of the skin. So next comes mucous membrane. Second one is now, even though skin covers the most of the parts of the body, there are certain orifices in our body. For example, the mouth, the nose, okay, and then we have eyes which are directly exposed to the exterior. So what happens is microorganisms or pathogens can enter through these orifices. So the lining of these cavities actually are lined with a lot of, they are lined with mucus. Okay. So there are cells that secrete mucus into the linings. For example, the GI tract, the respiratory tract, the urogenital tract, so these are all lined with mucous membrane. So also the GI tract shows peristaltic movement. Also, see, I forgot to mention this point. The most important part of the function of mucous membrane is basically to trap the pathogen. Okay, to trap the pathogen. Now, once the pathogen is tracked, the GI tract through peristalsis can actually expel the microorganism. Then the respiratory tract is lined with cilia. Okay, by cilia, by beating itself, cilia is nothing but hair-like projection. So by beating, it can actually get rid of the pathogens. Okay, then yeah. So then we discussed about, similarly the mucus in urogenital tract would also trap the microorganisms. Then also urogenital tract and also the GI tract, they also have microflora, okay, which is nothing but other commensal microorganisms or bacteria growing there, which would actually prevent the colonization of the invading pathogen. Okay. Then comes the the eyes and mouth, they constantly secrete tears and saliva, okay, which actually traps the pathogens and gets rid of it. Okay. So after discussing mechanical barriers, we'll go to what are the different physiological barriers or the chemical barriers that are present in the that is present in our body and provides immunity. Okay. So the second component is chemical barriers. Okay. So chemical barriers are actually of two types. So first would be low pH. Okay. Certain parts of the body are equipped with this low pH, okay, or they generate low pH, because of which the microorganisms will not be able to survive. Okay. And then there are antimicrobial molecules in our body. In our body that actually would fight against the invading microorganism. So we'll discuss these chemical barriers one by one. So first, we discussed about GI tract and the microorganism pathogen getting inside it. So the gastric secretion, let me write. So the gastric secretion actually from the stomach has a very low pH of about 1.5 to 2. So this makes the environment extremely inhospitable to the pathogen. Next we talked about skin. So skin has sebaceous gland that secrete sebum. Okay, sebum is nothing but a mix of different organic acids with low pH. Okay. With a pH of slightly greater than gastric juices. So this is pH is 3.0 to 5.0. Okay. So this retards the growth of microorganism on the skin. Then we talked about tears, okay, and mucous membrane. So all of the mucous membrane, the mucus specifically contains lysozyme. Okay. So where is lysozyme present? It is present in mucus, then it is present in tears, then saliva. Okay. So what basically lysozyme does is it breaks the cell wall of the bacteria, because of which the bacteria undergoes lysis and then it cannot survive. Okay. So this lyses cell wall of bacteria. Okay. Next we will talk about, I'm erasing this. So next we will discuss about the other enzymes So for digestion, different enzymes are secreted in the duodenum. Okay. So next we'll talk about so these are basically your lipases and proteases. Okay. So lipases break down lipids. Okay. So this will break down lipids, and proteases would break down proteins. Okay. So basically, be it any microorganism, they are basically composed of lipids and proteins. Okay. So when their outer surface, outer coating is broken down, so they basically they lose their ability to survive in the body of the host. Okay. Next comes so mother's milk. Okay. Mother's milk is rich in antimicrobial substances, specifically lactoferritin and neuraminic acid. So these provide immunity to the infant against invading pathogens. Next comes antimicrobial peptides. As the name suggests, these are proteins which are actually equipped with they are given the task of destroying the different invading pathogens such as bacteria. They work very efficiently against bacteria. Okay. Basically, these are charged in nature. The bacterial surface is negatively charged and these are positively charged, because of which they can attach to the microbial surface and then they can cause subsequent destruction of the bacteria. The example of microbial peptide would be defensin. Okay. Defensin. Actually the human skin is actually covered by 1 micrometer, okay, not millimeter, micrometer, 1 micrometer layer of defensins. And the last one would be interferons. Okay. Interferons. Now interferons are actually signaling proteins. Okay. So let's just say we have a cell which has been infected by a virus. So these virus infected cells would actually release interferons to the surroundings. So the healthy cells that lie around the virus infected cells, they would actually go on alert that they have to fight an incoming virus. Okay. Interferons, these are signaling proteins. Okay. So they alert viral invasion. Okay. So next we will discuss another component of innate immunity, which is phagocytosis. Okay. Once pathogens enter the by any means, if they are able to enter the skin, okay, so or skin, they are able to enter the skin or the mucous membrane. So so we have phagocytes, okay, circulating in the blood. So phagocytes can be viewed as security guards on patrol. So they are actually of different types. These are macrophages, neutrophils, so these would actually so they would surge to the site of infection, okay, or the site where the microorganism is spotted. They would engulf the invading microbe, thereby forming a large vesicle called as phagosome, okay. Then the phagosome would actually eventually, after they engulf the pathogen, the phagosome would fuse with lysosome. To form the phagolysosome. So the lysosome contains hydrolytic enzymes. So once after the fusion of lysosome with phagosome, the hydrolytic enzymes are secreted into the vesicle, and then the microorganism which is trapped inside it is destroyed, okay. So this actually leads to so now once it is digested, there would be a lot of substances which after the digestion there would be a lot of substances which are actually beneficial for the cell. So those would actually be absorbed, and the remaining which is there, that would actually be released. So phagocytosis is actually considered as the second line of defense, okay, the second line of defense. So next we will talk about fever. Okay. Now fever is actually a condition where the temperature of the body goes up, okay, temperature of the body goes up. So what happens because of this? So why does the temperature goes up? It is because of either endogenous pyrogens. Pyrogens are basically substances that raise the temperature of the body, okay, or this could be because of the bacterial toxins, okay. So fever is actually so the rise in temperature would lead to the destruction of sensitive pathogens. Fever is actually not a disease, but it is actually a sign that your body is fighting an infection, okay. And then the symptoms of the fever would actually help in diagnosis of the disease, okay, because of which the fever was caused. So next we'll talk about the next component, which is inflammation. Okay. So inflammation is basically the non-specific reaction of the body against an injury. So it is either directed against an injury or infection, okay. The injury could be due to mechanical, okay, due to cuts, okay, cuts or pricks, or it could be due to chemical agents, okay, chemical agents such as acids, alkali or venoms. Often bees and snakes, they sting and they cause this injury. Okay, so what happens basically at the site of injury? So we have mast cells. Okay. So mast cells actually, they secrete histamines and bradykinins. So these are actually vasodilators, which basically means that these cause the blood vessels, they cause the blood vessels to dilate, and the diameter of the blood vessels basically increases. So what happens? More blood is able to flow to the site of injury. Now, because of the more flow of the site of to the site of injury, macrophages also surge to this place. Okay, macrophages also surge to this place. Now what happens subsequently is that macrophages through phagocytosis would take care of the invading pathogen. Then and also because of the rushing of blood, we would have swelling there. Fluid accumulation would cause edema. Okay, because more blood is flushing to the area is generalized, generally it appears red in nature, reddish in color. Okay, so there are a few signs of inflammation. Okay, so these signs are so the signs are redness, then it is swelling. Okay, swelling is because of accumulation of fluids or when edema is caused. Then we have heat. So because of the inflammation, the area is actually has a higher temperature than the rest of the body. Then pain, okay, associated pain, and sometimes loss of function. Okay, so these are the signs of inflammation. So because of continuous, so we have macrophages in the site. So because of continuous phagocytic activity, a cavity is formed in the area. Okay, cavity is formed in the area. So this cavity actually here a lot of, so what this cavity basically consists of, so this has necrotic tissues. Okay, this has dead macrophages, and this has dead the excreted products which are expelled out of the body of the macrophage post the destruction of the pathogen. Okay, so in the cavity basically the fluid that is accumulated is called pus. Okay, and this pus formation goes on until the infection is suppressed. Okay, now coming to the next component of innate immunity, which is acute phase proteins. So this is number F. Acute phase proteins. Okay, so these proteins are they are secreted or they are produced in response to cytokinins, inflammatory cytokinins. So like your interferon, cytokinins are basically signaling molecules. So these cytokines can these cytokines actually these invoke or they compel the liver, okay, to produce this acute phase proteins. So this acute phase, the concentration of the acute phase proteins can go up or down by 25%. Okay, so these can either go up or down by 25% in response to the cytokinins. So these are, so some of them are actually called so positive acute phase protein. In short, these are also called APPs. Okay, so these can be positive APPs and negative APPs. Okay, the basic task of this acute phase proteins is to suppress viral infections. Okay, to suppress viral infections. So during so the whole reaction is called acute phase reaction. Okay, so this reaction involves fever. Okay, and this leads to increase of circulation of many phagocytes in the blood. Okay, so next and the last component of innate immunity is actually natural or NK cells. Okay, so these are a type of non-granular non-phagocytic what does this term means? So these are non-phagocytic in nature. So basically they won't exhibit any phagocytosis. If they do not phagocytose, how do they work? How do they destroy the pathogens? The answer lies in this granular part. So basically they contains substances called as perforins and cytolysins. So these appear in the cell in the form of granules. So perforins are actually pore forming proteins, and cytolysin is toxic to the cells, okay, or toxic to the invading pathogen. So what happens is that, so when these are released, they ultimately lead to lysis of the cells. Okay, now what cells are we really talking about here? So the different cells are actually and natural killer cells, these are again not specific. Okay, any component of innate immunity if we take are not specific. What do we mean by that? So natural killer cells have the they have the ability to destroy tumor cells or virus infected cells through these two proteins, perforins and cytolysins. Okay, so this was all about the innate immunity. So the next topic that we will discuss is acquired immunity. So the next type of immunity is acquired immunity. Okay. Now, as the name suggests, this immunity is not present since birth, and this is acquired, which basically means the body learns this kind of immunity. How does it learn it? When it encounters different pathogens, okay, it develops means to fight it. Okay. So this actually, this learns to fight against pathogens. Now it not just learns, it actually finds out the best way to kill a pathogen. Okay, so therefore it adapts itself. Okay, therefore acquired immunity is also referred to as adaptive immunity. And then the response towards pathogens is very specific. Okay, thus it is also known as it is specific immunity. Okay. So usually what happens when the immune system is exposed to the pathogen for the first time, so a primary response is generated. Okay, so the primary response is normally not very effective, but however after the primary response, so let's just say it was able to be able to get rid of the microbe, and then when it encounters the microbe for a second time, it actually a secondary response is generated. Now the secondary response is very efficient, okay, and very fast. Okay, so within a brief period of time, it can actually get rid of the entire infection. Yeah. So this acquired immunity is actually conferred by lymphocytes. So the different lymphocytes present in the body are B or T lymphocytes. Now what does this stand for? So T stands for thymus. Okay. So thymus is the place, is the organ from where these lymphocytes are derived. And then B stands for, even though B lymphocytes are generated in the bone marrow, basically this B stands for bursa of Fabricius. Okay, so this is the lymphatic organ from in the birds from where this B lymphocyte was first isolated. Okay, this is named after, B stands for bursa of Fabricius and not bone marrow, even though in human body the B lymphocytes are derived from bone marrow. Okay, so these actually confer acquired immunity. Now let us talk about the different types of immunity, types of acquired immunity. Now acquired immunity, there are two types. Natural acquired immunity, okay, which is acquired through continuous exposure to different pathogens, and then there is artificial acquired immunity, where this immunity is obtained through vaccination, okay, by or administrating different toxoids. Okay, so natural immunity. So I'm really sorry, I missed a term. So this here we are actually talking about the different types of immunity are active acquired immunity, which consists of natural and artificial immunity, and then we have passive acquired immunity, which again consists of natural and artificial immunity. So what do we mean by active natural acquired immunity? Okay, so this actually is caused by continuous exposure to pathogens. Okay, now artificial is caused through, so because it is active, the body is actually generating this kind of immunity. Now passive means this is actually transferred to the individual. So now the natural passive acquired immunity is actually transferred from mother to fetus. Okay, So there are two ways by which mother transfers it. So first is the antibody IgG. So in a brief, so in the next class we'll actually be talking about in a short while we'll be talking about antibodies. Okay. So IgG is actually transferred through placenta. Okay. So this IgG confers protection to the fetus for 6 months. Okay. And the next type of antibody that is transferred from mother to fetus is IgA, okay, which is actually present in mother's first milk or colostrum. Okay. Then comes passive, artificial passive acquired immunity, where again, so antibodies, okay, antibodies or antisera. Antisera is basically serum containing antibodies. Okay. So antibodies or antisera is transferred from one individual who is immunized to another non-immunized individual, okay, where thereby conferring immunity to it. Okay. So example of this artificial passive acquired immunity is, one of the examples would be your ATS, okay, or the anti-tetanus serum. So the characteristic of these, the passive immunity, is that this is valid for a limited time period. Okay. So short period of time. It lasts for a short period of time. Okay. So now, having said this, we'll talk at length about the different mechanisms by which this active natural acquired immunity works. Okay. So yeah, so there are two ways in which, so let me write it as active acquired immunity. Okay. So there are two ways in which it works. So the first is called cell mediated immune response. Okay. Now, what is cell mediated immune response? So remember, we talked about two types of lymphocytes that are involved in acquired immunity, B lymphocytes and T lymphocytes. Though this here is conferred by T lymphocytes. Okay. Okay. Now, so here what happens is that, so T cells respond to T lymphocytes or T cells. These responds to different type of pathogens. Now what happens is that once they come in contact with different pathogens, so they become activated. Okay. Let me write here. They get activated when they come in contact with different pathogens. They undergo proliferation. Okay. Proliferation. So these T cells are basically naive. Okay. These are naive, which means they are innocent. Okay, nascent. So these, when come in contact with pathogens and they are activated, they undergo proliferation. Proliferation is nothing but a rapid increase in number of T cells. Then they would actually differentiate into different types of T cells. So what are the different types? So these are T cytotoxic cells, or we write it here, TC cells. So these cells would directly go and kill your virus infected or pathogen infected cells. Okay. So then comes T helper cells. T helper cells. Okay. So these cells they actually help the B lymphocytes to produce antibodies. Okay, we'll discuss in a brief while what are B lymphocytes, what is the role of B lymphocytes, and then there are T memory cells. Okay, or T memory cells, which actually, so you remember how we talked about primary response evoking a smaller immune response, and then the secondary response being much effective and much efficient than the primary response. This is because of this T memory cells. So these are cells that retain the memory of the invading pathogen, so that the next time they encounter it, the body can evoke a heightened response towards them. Okay. So this is all about cell mediated immune response. The next kind of immune, next type of active acquired immunity is by the mechanism of humoral immunity. Okay. So next is the second type is humoral immunity. So this is conferred by our B lymphocytes. And this is mediated by antibodies. Okay. So basically B lymphocytes are the ones that would secrete antibody, but how? Okay. So first step here is when B lymphocytes come in contact with the pathogen, so they are activated. Okay. So activated B cells would actually take by the help of so these would actually transform into, so plasma cells are very big, way bigger than your activated B cells, and this transformation, so these are very big, and these are the cells that actually secrete your antibodies. So this transformation is brought about by the help of T helper cells. Okay. This is where T helper cells help the activated B cells to become plasma cells, and now these plasma cells would actually secrete, so let me write it here, these plasma cells would actually secrete antibodies. Okay. Antibodies are basically, let me write it here, antibodies are basically glycoproteins. Okay. Are basically glycoprotein. So if you see, look at the word, there are two components, glyco and proteins. Proteins we know about it, glyco is the carbohydrate part. Okay. So this is a conjugate of carbohydrate and proteins. So what happens here is that, so there are different classes of antibodies. Okay. So 5 classes of antibodies are present in our body, which are IgM, okay, IgG, IgA, IgD and IgA. So in our next class we will talk about the structure of antibodies, the comparison between what are the difference between different antibodies, and then we will talk about antibody antigen antibody interaction, and then we'll shift to the different diseases that are caused by pathogens. Okay, that's all for today's class. Thank you.